goodbye, my love. I have to find more reasons to live. Waiting for a call, for a breath, but I hope it's over. Goodbye, and I feel inside a ray of light. Life is getting around me, and I know that's the start. My life is dancing around me, and I feel that's my start. There is something new down in my heart. It's a new beginning. Goodbye. Now I say goodbye to you, my love. Life is getting around me, and I know that's the start. My life is dancing around me, and I feel that's my start. There is something new down in my heart. It's a new beginning. Goodbye. Now I say goodbye to you, my love. Life is getting around me, and I know that's the start. My life is dancing around me, and I feel that's my start. Life is getting around me, and I know that's the start.
Hello, welcome to our fifth webinar, or we called our Zebinars, during our month of February. We have several others coming up too, all on the theme of a diagnostic odyssey. So I'll have a couple of slides to show you as an introduction to this theme. We call our month of February, obviously, as you know, because of zebra is a symbol of rare diseases, and February is the Rare Disease Month because of International Rare Disease Day on February 28th or 29th. So there are about 7,000 or between 7,000 and 8,000 rare disease, which makes it difficult to get a diagnosis often rapidly. A brief introduction to the Regroupement Québécois de Maladies Orphelines, which we call the Quebec Coalition of Orphan Diseases in English. Our mission is one of informing, educating, promoting research, and also advocating for rare disease plan, strategy, or programs, but we also have, we operate our CARE4RARE Center, which is an information and support center for any rare disease and for undiagnosed diseases. So we help out with information on any of the rare diseases. We can send people some resources and references in the medical system. So for the undiagnosed, we can help them out by help them navigate the health system and find the right place for the diagnosis, and sometimes it works out. So just some statistics. Usually we say that the percentage of people with rare diseases in the populations is between 6 and 8%. So in Quebec, that would mean about 700,000 people that are affected or, let's say, concerned also by a rare disease. As you know, many are hereditary, with several people in the family that are concerned, example, carriers. And that would include about 500,000 children. And we say, this is the estimate we always hear about, that at one point in time, there would be 40 to 50% of people with rare diseases are without a diagnosis. Now, some of these will obviously finally get a diagnosis. Surveys around the world, and a survey that we did some years ago, statistics are pretty much similar. It's about the different surveys would say that it's the average number of years to get a diagnosis, somewhere around 7 or 8 years. In our survey, 30% of the respondents said that it took 3 years or more to get a diagnosis, 18% more than 10 years. 30% consulted 5 doctors or more before getting a diagnosis. So there are consequences for these delays in diagnosis, obviously medical, because sometimes they're along the way and during their diagnostic odyssey, there are some false diagnoses, and it's difficult obviously to treat. So sometimes it's treating by trial and error, and sometimes, well, there are some inappropriate treatments or interventions, and especially for the genetic, possibly hereditary disorders. Well, you can't do a specific genetic counseling, or sometimes there's no genetic counseling at all. So it's difficult to know the risks for other family members. And obviously there's some psychosocial consequences of this diagnostic odyssey. The uncertainty at many levels, but uncertainty also about the prognosis of the disorder, its progression, its evolution. There are some false hopes when you go through different possible diagnosis. There's difficulty accessing some services in the health and the social services in our health system. Obviously, there's some psychosocial impact. There can be a lot of distress and loss of confidence in the health system, especially for those who are not believed, because that happens sometimes when it's a certain type of disorder with non-specific symptoms, or an invisible rare disease. Sometimes doctors will bring up the hypothesis that it's psychosomatic or psychological, even psychiatric. So some people lose confidence in the health system. So as I said, there are between 7,000 and 8,000 rare diseases, and physicians, doctors, health professionals cannot learn all these diseases, but there are some solutions that I think we can implement to help reduce the delay in diagnosis. In France, a country that has a rare disease plan since 2005, they talk about the culture of doubt. So bringing physicians to doubt, to think that it could be a rare disease, that they can think of a rare disease, even if all tests and exams came up negative and they can't find an immediate diagnosis, but to doubt and to consult. And for this, we think, and that's what we're asking in rare disease strategy here in Quebec, and I guess it applies to across Canada too, is to have a specific training of physicians and other health professionals about rare diseases, not learning the 7,000-8,000, as I said, but an approach that they understand what it is to have a rare disease, the impact on the families, the special needs of people with rare disease, and especially a diagnostic approach. But they would also need resources. So first-line physicians, family physicians, those in the emergency services, other health specialists, they should have resources to know where to refer their patients, where are the experts for some categories of disease, so that we can reduce that delay in diagnosis. These are two examples of personal stories that are being posted on our Facebook page during this month. And they are two examples of results, positive results to get a diagnosis through the new technologies, sequencing technologies, which Kristin, the researcher presenting today, will address. So this person got her diagnosis at 33 years, a very, very, we can call ultra-rare disease, and the gene was identified through exome sequencing, and she has only 2 distant cousins that also have her disorder, and somebody else reported elsewhere in the world. So they are very, very rare. But in some cases, we can get the diagnosis by these technologies. And Thomas here has just he has just got his diagnosis very recently, 14 years old. And he too it's a disorder associated with the gene, and the kind of new diseases that were difficult to diagnose because they presented either something that hasn't been seen before, presented differently. So now they have at least a gene name associated with the disorder. In the case of Marie-Josée, her disorder has an acronym, CAGSSS, which describes the disorder. So we'll be hearing our guest today, Kristin Kernohan, which will give us other examples of family that finally got diagnosis through these technologies. And before, I want to thank our partners, our sponsors, for all our Month of Zebre activities, and for some of our projects at the RQMO. And before I present our presenter today, I want to tell you that you can write questions during her presentation, or and when she's finished, in the Q&A at the bottom of your screen, and for the francophones who are getting translation, in the Q&R at the bottom of their screen, to any questions to our guest today. So I'll present Dr. Kernohan. She's a senior scientist at Children's Hospital of Eastern Ontario Research Institute, but she's also on the clinical side as the clinical doctoral scientist at the Newborn Screening Program in Ontario. She has a BSc in specialized in genetics and biochemistry from the University of Western Ontario. And she did a postdoctoral fellowship in this project, CARE4RARE. And she's a certified Canadian College of Medical Geneticists in the molecular genetics and is why she's joined the newborn screening Ontario program. Her, obviously, and she'll be talking about this today, her research interests are the identification of new disease genes and new technologies for molecular diagnosis. So I'll hand the presentation to Dr. Kristin Kernohan.
Thank you. Thanks very much for the invitation to share today with you my work and that of many others in Canada. So certainly I'm here speaking on behalf of a very large group. And so I'm going to talk to you today about CARE4RARE Solve, which is a research project here in Canada. It's a pan-Canadian project. As was said, I have two hats. I'm a clinical scientist, but also I'm involved in translational research for rare diseases. And so this is my research work that I'm going to speak about today. And so CARE4RARE Solve is looking at using new technologies to deliver innovative diagnostic care for rare genetic diseases in Canada. Now, this did start off and even predates my involvement in the project with something called Forge Canada. Forge then in the second iteration of these large-scale 5-year grants, the second iteration was called CARE4RARE, and we're now in the third iteration, which we've called CARE4RARE Solve, and it's actually coming to an end, and we're looking towards the future. And so some of my next slides actually overlap a little bit with what Gail presented, but that's okay. We'll just recap a bit. So rare genetic diseases. There's about 1 million Canadians who are impacted by rare genetic diseases, and the majority of these are children. There are about 7,000 rare genetic diseases described to date, but we're anticipating that there's about 5,000 at least that we've yet to discover. And certainly that number is highly debated and is kind of a moving target of really trying to understand what is the breadth of genomic disease that we haven't yet identified. And so certainly the majority of rare disease patients, unfortunately, are undiagnosed after what was traditional genetic testing. And I'm going to talk today a little bit about what's evolved in the last 5 to 10 years and where we're looking at for the future. Okay, so I first wanted to briefly talk about the concept of discovering a disease gene. And so if you look here along the bottom, and certainly it's a bit of an outdated graph, unfortunately there's not a current one available, but it gives you an idea of as we started to develop genomic technologies, and this predates my existence on Earth, but in 1986 you can see there's just a few diseases that were discovered, and this was using technologies that were really labor intensive to identify. And as some new methods evolved and things became easier, we started to discover more and more conditions. And then in the early 2010, and just before that actually, we started to be able to use a new technology called next generation sequencing, or NGS for short. And so this has really revolutionized genomics and genomic medicine because it allowed us to sequence DNA very, very quickly and inexpensively compared to the past. To give you an idea, if we want to sequence just one gene with methods that were used in the past, it might take several days just to sequence one gene, whereas now we can use this next generation sequencing to sequence your entire genome in that much time. And so there's about 3 billion bases that we can get through in about the same amount of time we used to be able to get through in about 3,000. And so it's really just made a huge difference in speeding up our pace of understanding the genome and being able to identify these new disorders. Okay, so I talked about these traditional genetics, as we call it, and really at this scale, we're checking one or a few genes. And certainly for some patients, this is actually still the appropriate way to do it. If they present with symptoms that are highly consistent with one condition, then it makes sense to go do that. But for a lot of rare disease patients, the spectrum of their condition is such that we really can't pinpoint exactly what that might be. And so because of that, we needed a better test. And so exome sequencing, as it's called, has been that. And exome sequencing, in comparison to traditional genetics, actually checks all about 20,000 genes that are in your genome. And so in all of our DNA is these about 20,000 genes. And by being able to look at them all at once, this really is what's greatly improved our ability to obtain a diagnosis in rare disease patients. So exome sequencing really is the best test we have ever had for rare genetic disease. And some of the similar statistics to what Gail gave at the beginning On average, before, rare disease patients would often see between 3 and 5 specialists. About 40% received greater than 3 misdiagnoses. It took on average 3 to 6 years to obtain a diagnosis, and in terms of cost to the healthcare system, averaged about $15,000 per patient. Now, with next generation sequencing, we're able to obtain a diagnosis between about 25 and 60%, and so that's quite a large range. But that range is actually based on the clinical indication. And so certainly for some symptoms that present, so children with multisystemic involvement and neurological features, the diagnostic rate is much higher than adults, for example, with only one symptom, which may or may not be genetic. And so with this, it's certainly been a great success in the genetic disease field that we're able to provide answers to significantly more families. Over 200,000 rare disease patients now are clinically sequenced worldwide, and this allows us to provide informed care and really to help improve family well-being. Now, I will say that I have photos of some of the families that are participating in our project throughout, and these are shared with their permission, as they're quite happy to tell their story. And so, again, we look at traditional genetics, one or a few genes. Exome sequencing, we can check all 20,000 genes. So what can we do with an exome then if we've sequenced all the genes? First off, we can find diagnoses in known disease genes. So as we said, there's about 7,000 that have been described to date. And by sequencing these genes, we can then look for mutations in these that may cause disease. If there's no answers there, we can also find potential new disease genes. However, what we know is that exome sequencing is not perfect, and it does still miss things. So why do exomes miss things? Just a quick kind of recap of biology. So what happens in our cells, we have our DNA, and this codes for what ends up being the structural and functional products, which is proteins. And so to get from DNA to protein, we have some messaging steps in between. When we are looking at an exome or looking at any DNA sequence, we're looking at your DNA and trying to predict what a change might do to the end product. If we then look at, well, what is an exome sequencing, and look at a little bit more about the structure of your DNA and what does it look like. So we have those about 20,000 genes, but the actual functional region of those only makes up about 1% of our total DNA. So you can see here on the right, I have a chromosome, which is our large chunks of DNA. We all have 46 of them. And we have our coding regions, and then in between that, it's actually 99% of our DNA that doesn't make proteins. The rest of this is filled with instructions, it's space filler, and really we're actually still learning what this is. It's often called genomic dark matter, and we don't fully understand what that space does. But certainly of what we do understand, there's instructions for how to make proteins, for where to make those proteins, for when to make them, and that's really important for how our genome functions, and certainly for genetic disease. And so exomes are really good, but if we look at our analogy, if we now have a whole page of a book, we just have one word that's our coding DNA, and we're going to look at all of the other words to try to understand what that means. And so we need new technologies to help us be able to do that. Okay, and so this is what led to CARE4RARE Solve, and our overall high-level goals, we have two. So the first is to fully understand the biology of unsolved rare diseases. And the second is to facilitate timely diagnosis and optimal healthcare delivery of genome-wide sequencing. And so CARE4RARE Solve is a very large team, as I said. I'm here on behalf of many, many individuals, and there's 21 sites across Canada. It's led from here in Ottawa. The two lead PIs is Dr. Kim Boycott, who's here in Ottawa, and Dr. Mike Brudno, who's in Toronto. There's the 21 sites across Canada. We have 100 physicians, 50 scientists, and over 350 collaborators, and those numbers are growing all the time. And so certainly this work has, the large-scale collaborations is really what's allowed us to be successful in trying to move the care for these rare disease patients and families forward. Okay, so CARE4RARE, we have broken down into 4 main activities. I'm going to walk you through each of these today. And so the first is to optimize data collection and analysis. The second is looking for new disease genes. The third is developing innovative approaches. And the fourth is to optimize delivery. And so I did, before I get into each of these activities, want to just quickly mention how did the patients and families get involved in our project. And so CARE4RARE has what we call inclusion criteria for healthcare providers. If a rare disease patient has had exome sequencing on a clinical basis, which is non-diagnostic, their geneticist can then assess whether they meet the inclusion criteria to apply to the program. And so our enrollment, as I said, is wrapping up as we near the end of CARE4RARE Solve, and certainly we're looking to the future in terms of obtaining new projects, funding, and what we would like to do next. And so I mentioned this in our inclusion criteria, and why do we have this and what does it do? Well, it aims to ensure that the condition in question is in fact genetic, and that samples are required are available. So certainly there's lots of families out there with genetic conditions, but unfortunately if the individual is deceased or is in a different country that we can't get samples from, unfortunately we know we're not going to be able to help that family and to try because we don't have the samples we need. And likewise, we only have, as we say, limited amounts of resources, as I'm sure, and so we tried to limit the incorporation into our project of conditions that are suspected to be genetic for obvious reasons. Our assays really wouldn't help anyone who doesn't have a genetic condition. Okay, moving into the first activity. So when we sequence someone's DNA, this creates tons and tons of data. We then have many fancy computer scripts and algorithms and programs that help us look at that data and decide where are the mutations and whether these mutations can potentially contribute to disease. But if we're looking at this individually, it's actually not as optimal as if we were able to look at these together. And so what I mean by this is that if we have a patient in Ottawa who, for example, has a seizure disorder with a dysmorphic features, and there's a similar patient or group of patients across the country out in British Columbia, well, it actually is better if we can look at their DNA together sometimes and try to look for genes that have common changes in them. And so being able to share data both within Canada and internationally can help us find more answers for patients. But before we started CARE4RARE Solve, all of the data was actually just, as we call, siloed across the country at local centers, and we didn't have the ability to share that. And so one of the first main things that CARE4RARE Solve we did was create a database that we've called Genomics4RD to be a pan-Canadian data set for rare disease patients. And so all of the families and patients who've participated in CARE4RARE Solve have consented to have their data shared in Genomics4RD. It's anonymized. And that we are then able to look at cohorts of samples. So to give you an idea, we have some conditions with up to 30, 40, 50 people who have what looks to be something similar, or smaller groups, certainly. And this also, in the future, will allow us to work with collaborators worldwide. And so we've now established that data set and have many patients already, hundreds of patients that are already in there. Okay, so the second activity I wanted to talk about is looking for new disease genes. So when we analyze someone's exome, we look first at known disease genes. So you always hear the phrase, when you hear hoofs or footprints, think horses, not zebras. And this is actually, I didn't mean this to align with your theme of February, but this wonderfully aligns. And so the first thing that we would do is look for variants in known disease genes that might explain the patient's condition. If we can't find anything, this is where then we would start looking at what could be potentially new disease genes. And this is where we flip to zebra mode. Okay, so how do we identify new disease genes? The first thing we would do is look at the DNA changes that are in that person. So everyone has thousands of DNA changes that are different from one another. The vast majority of these don't cause disease and are not harmful at all. It's what makes us all unique as individuals. But it's a bit like searching for a needle in a haystack, where we need to identify which of those DNA changes are actually causing a person's condition. And so in doing that, some of the things that we look at is that same DNA change, or we often call it a DNA variant, in unaffected people in the family. So if I have the same change as my brother and he's healthy and I have a condition, well, chances are that that's not the DNA change that's causing it because he doesn't have the condition. And so we can use testing other people in the family to help inform whether that DNA change might be causing that condition or not. The other thing we would look at is outside of families, we have access to very large control cohorts. So there's hundreds of thousands of people whose DNA has been sequenced and anonymously compiled in data sets that we can look to see how many people in a healthy population have the same mutation. Again, with the same thought of, well, if this has been seen hundreds or thousands of times in people who don't have a genetic disease, then chances are that's not the right variant. The next thing we would look at is we have a number of fancy computer algorithms that help us look at whether a variant would potentially damage a protein's function. And we do that by looking at different characteristics. A couple things include evolution, so has it been conserved over time? If that same DNA base is the same in many, many, many species, then chances are it's more important than if it's different across different species. The other thing we can do is just look at the structural characteristics of what that building block looks like and see if it might damage by swapping in something else. If a DNA change looks that it might be potentially disease-causing, or at least have an ability to be based on frequency and other things, we then would take a look at the gene to see, first of all, is the gene expressed in the right place? If we're looking at a family, for example, with a kidney condition, and we have a gene that's only expressed in the brain, well, that likely doesn't make sense to be the disease-causing gene in that family, and so we're probably not looking in the right place. Similarly, does the gene function make sense? If we have found a mutation in a gene for brain development in a child with developmental delay and brain abnormalities, perhaps, then that would make a lot of sense. But again, if we go back to the kidney analogy, if it's a gene for brain development in a family with kidney disease, well, that doesn't make sense. In addition to that, there's often other diseases associated with the gene. So sometimes different types of mutations in the same gene can cause different conditions, but they usually affect the same system. So if it's a neurological disease and we have something that's a little bit different with a different type of mutation, but still a neurological disease, well, that can also help tell us we might be on the right path. And then finally, there's a lot of emerging evidence in what we call model organisms. And so people have looked at inactivating different genes in mice and in fish models and flies and other animal models, so we can look if those genes are important for development. And so that can help tell us also which systems in the body that perhaps a gene is important for and whether we're looking in the right place or not. So we've identified what we think might be a DNA mutation in a new gene that causes a disease. How do we go about confirming this information? How does it get from my one computer with me looking at it to being a confirmed disease gene that people around the world could then look for in their patients? And so the key for this is that we need multiple similarly affected unrelated individuals with variants in the same gene. So we need to find more families around the world that have the same thing to be able to confirm that this truly is that. In addition to that, I will say we also will often do studies that we call functional studies. We look at that protein's function in cells from that patient in the lab, and sometimes we'll even actually put that same mutation into a model organism. So we can make fish with specific mutations or mice or other animal models. And so once we have a number of affected individuals who are not related, this is how we're able to confirm a disease gene. How do you find those other individuals? And so this is one of the really tricky parts of rare disease that we've been working to solve. And so this is our what we call N of 1 problem. And so here on the left, we have patient 1 with our clinical geneticist. This is Kim Boycott, who's the lead of CARE4RARE. And she's here in Ottawa and has a patient with these features and a number of genes that she's interested in. She can then call up a friend, is how we used to do this, and say, you know what, I have this friend, Hans, in Europe. Hans, do you have anyone who's similar? And sometimes this would work. And so certainly in this case, Hans then had a patient with the same gene or mutation in the same gene that had a lot of similar features. And this is how, for many years, we went about finding families. But I'm sure, as you can imagine, this is not the most efficient way, always calling up a friend. And so with this, the rare disease community as a whole across the world, and we've been leading this as one of the groups, has developed a computer resource to do this for us. And so we call this, it's called the Matchmaker Exchange. And you can see that we're one of the top left here, CARE4RARE, a node of this or a group of this. And so this is groups worldwide who are also working on identifying new disease genes. And those genes are then submitted through the Matchmaker Exchange. And so how it works is I would submit, here's the gene and just high-level phenotypic information. So perhaps I have a child with neurodevelopmental delay and seizures, and I have a mutation in gene X that I think is causative. I would put that information in. You'll note there's nothing that's personal health information or identified. And if that then matches with someone else has put in the same gene, it will send us each an email to say, hey, you should go talk to each other. You've now matched on this gene. And to give you an idea of the scope, CARE4RARE, we've now submitted 194 novel gene candidates to the Matchmaker Exchange. This has resulted in 1,514 matches, so often multiple groups have hit the same thing. To give you an idea, we found a mutation last year, put it in, and we hit 33 different groups at the same time. And so clearly many people had discovered this, and so we can get together these large cohorts of patients, and then that allows us to compare the phenotypes and really define the clinical spectrum of what that new condition is. And certainly it's not perfect. A lot of these don't pan out, but about 15% of them result in collaborations, meaning that we think about 15% are real. So sometimes they're not what we call real, meaning we'll match with someone else, but their patient has a very different condition than ours. And so that doesn't confirm that these are the same. Sometimes it doesn't convince us that we're not right, but we keep looking at that point. To give you an idea of the breadth of international connection within the Matchmaker Exchange, there's now patients and contributors from 67 different countries worldwide. There's over 44,000 patients, 3,000 contributors, and over 5,000 cases are shared a week. And so certainly this is a very powerful tool for us to be able to identify additional patients with the same condition to confirm that we're right and that this truly is a new condition that should be described in the literature. And I just wanted to end up talking about novel gene discovery by just telling a little bit of an example. And so this here is a little girl who presented to us with global developmental delay. She had troubles eating, and so was G-tube fed, and a number of other difficulties. Now, genetic testing that was done on a clinical basis was negative, so she had clinical exome testing. We then took that data back in the research lab and we re-looked at it again, and we agreed that there was nothing in known disease genes. And then following that, started taking a look at novel disease genes. And what we identified was what we call a de novo variant, meaning that it wasn't in her parents, it's only in her, in a gene called CEPT11. And so we then entered this into the Matchmaker Exchange and matched with a group in Europe who had a very similar patient. And so this is now being prepared to be presented in the literature. Certainly I'll also say that often once genes are in the literature, we'll start to see more and more patients receiving that diagnosis because that's out there for now for other clinical labs and groups to look at. Okay, so in total, CARE4RARE has now identified 203 new disease genes, and we are actively studying just about 1,900 families. I'm sure you can imagine that lots and lots of patients. We have tons of data that we're looking at and really trying to actively study all of these families to identify what these new genetic conditions are. Okay, so I talked a little bit about matchmaking and about exome sequencing, but I also mentioned that we are a little bit limited in our current approaches, both for exome sequencing, and I'll now highlight some of the issues with matching. And so the next activity of CARE4RARE is actually looking for new approaches. How can we improve on what we've been doing? What we've been doing clearly worked for a lot of patients and families, but not for all. And so we know that there's room for improvement, and that's what we're looking to do. And so in the matching that I just talked about, both groups, so at the top here, I have two-sided matchmaking. So for this matching to work, both groups had to have identified that same disease gene as what they think is causative. If, for example, the second group hadn't, then we wouldn't have matched. And so one of the things that we're working on moving to is what we call one-sided matching, where we can go into our data set, so into Genomics 4D, for example, and say, I want to see any patients with this phenotype and variants in this gene. And so within our Genomics 4RD set, this would then tell us if there was a similar patient in Canada and allow us to look at that to see if it might be causative there. And so this one-sided matching will eliminate one of the barriers, which is that both groups had to have identified this. And sometimes it's easier to identify in some patients than others. But if you know what you're looking for, like in this data pool, then we might be able to identify that second patient. And so this is one of the new approaches that we're looking at developing. The next is using new lab technologies. And so this is certainly where I focus a lot of my time. I talked about how exome sequencing, we're only sequencing these coding genes in the genome. And so that's about 1%. But the other 99% is very important too. We know at least 15% of mutations are likely going to be regulatory, meaning that we wouldn't pick them up with exome sequencing outside of that region. And so we're now using what's called whole genome sequencing, where we sequence the entire genome. And so it's a lot more data. But in doing that, that will allow us to pick up some of those mutations that we would have missed with exome sequencing. The other thing that we're doing is using a number of new technologies to look downstream of DNA. So I mentioned how we have DNA and it encodes for proteins, and we have a number of messaging steps in between. And we're looking at the DNA and trying to predict what might happen downstream. We're now able to use some new technologies that can actually just look downstream. So we can look at all the proteins in your cell, or the message. So this mRNA is actually the messaging step in the middle. We can look at your mRNA, we can look at your lipid profiles, for example, to try to pinpoint where in the DNA we should be looking for those mutations. Okay, and then the final activity is looking at optimizing delivery. So as I said, we're working on all these new ways to diagnose rare disease patients. And in the end, we want to be able to help transition this into clinical care, but to do this in an informed way so that we can help the most patients. And so as part of this, we're recruiting a prospective cohort in which we're analyzing clinical utility. So how many of these patients received a diagnosis, comparing the cost to diagnosis with what the clinical care would have been prior to new technologies, and then developing new ways to consent patients so that it's a better experience for everyone. All right, so in conclusion, CARE4RARE aims to improve the diagnoses and lives of rare disease patients by using new databases and patient matching to help data analysis, developing new technologies to assess new regions of the genome, identifying new disease-causing genes, and aiding in clinical delivery of genomic technologies. And with that, I just want to end by thanking the many patients and families for participating in our study, as well as all of the different funding and collaborating organizations. And I'm happy to answer any questions.
Well, thank you very much, Kristin, for this overview of this very, very broad project that has given results to many families, and I think with what you're starting to do now with more than exome sequencing will be giving hope to a lot of other families too. So this is in a research context, and just at the end you talked about the clinical utility, but I've heard that in Ontario they're already advanced, I think, in offering exome sequencing in the clinical level. Can you expand on that? How is it available? Are there special criteria for the families?
Yeah, for sure. So exome sequencing has been available to Ontario patients for many years now. There is criteria through the Ministry of Health. It used to go out of country. More recently, actually, there is an initiative to bring the sequencing back to Ontario. And so it's being run, there's a collaboration between Ottawa and Toronto, and the sequencing is being done and analyzed between those sites. And this is part of this project looking at what is the diagnostic rate of sequencing here in Ontario and gathering the information that the Ministry of Health needs to assess really what is the best path for those rare disease patients. And part of the evaluating that is that activity 4 that I spoke of. So yes, it is, exome sequencing is clinically available for Ontario patients and has been for many years now. There is strict criteria that have been defined in terms of who is most likely to benefit from that test. And as I said, it's now moving in province where it's been out of country for until now.
Okay, because I don't know if you've heard, but we're in this year, in 2022, we're supposed to have our network for molecular diagnosis, clinical diagnosis in Quebec, and we to do more genetic testing within the province instead of sending it out. And we're hoping also that clinical exome sequencing will be available too. But
That's wonderful.
as you know, it's also available in research in some centers in Quebec.
Yes, for sure.
You talked about genomics for RD, which is really the principle of sharing this data. And I don't know if you saw a kind of a letter editorial in Nature in the last week. There was an article just about that, the necessity of sharing internationally all this data. Actually, I thought there was already a lot of sharing because of ClinVar and Matchmaker that you, but I found out that it's not still not open access to all this data to all researchers. So I guess you're working also, especially with EARDIC, the Consortium on Research for Rare Diseases. I guess your group is really, I guess, promoting open access to all and sharing of all this data.
Yeah, it's an interesting problem essentially because we want to be able to share information to help people, but also to respect patients' privacy. And so without having all of their genomic data available for everyone. And so that's one of the things that Genomics for RD has really been working to accomplish, where it's a protected data set, but researchers can essentially apply to us to say they want access to certain levels of data, and depending on they would then sign confidential agreements, et cetera, and that they're going to use this data in line with our ethics. And it would allow either of them to look at cohorted data sets, so not on a patient-specific level or on a patient-specific level, depending on what they need to do. It's anonymized data, but it would allow them basically to ask questions to help answer their patients with our patients, so hopefully providing an answer for both sides. And so that's one of the things with Genomics for RD that we aim to do, is just provide a responsible way that protected patient privacy, but that would allow us to help get answers for those families and help move the rare disease field forward so we can provide answers for more families worldwide.
Okay, and I guess there's some obstacles are the fact that different countries or continents like Europe and the United States have different regulations for privacy of data, genetic, especially genetic data, I guess.
Very much so, yes. And everyone has their own ethical frameworks that we want to make sure that we abide by. And so that's why, as I said, patients are, as they consent into Genomics for RD, it's very clear in terms of what data would be shared and what is not, and they can decide if they're comfortable with that or not. And certainly I think the response has been very positive, overwhelming, and these families are looking for answers and wanting to help find answers.
Yes, I think most of them are very happy to be enrolled in such a project and are happy to share their information. I don't know if there's questions from my audience. I haven't seen any yet, so I'll wait for some, but I have really more genetic questions and about polygenic diseases. Is that something that you can suspect sometimes in your data? Because obviously there are some disorders that are probably, related to more than one gene, and even more with external environmental factors. And I'll give you an example because it's something we discuss a lot. I don't know if you're aware about Ehlers-Danlos syndrome hypermobile type, which is the only type of this syndrome for which we haven't identified a gene yet. So everybody's suspecting that there's more than one genes and other factors, and there's work being done on this internationally at this time. But is it something you can suspect sometimes when you look at your data, or you have all these variants and you don't know, is it one or is it two, is it three? Or
I would say I don't have a great answer because we're not quite there yet. So we're still, as I said, trying to figure out the monogenic disease one. And certainly I think that's where the future will be. And that's one of the great challenges that we need to tackle, is to really figure out these polygenic ones. There is many research groups that are starting to look at that, and they have polygenic risk scores, et cetera. I would say that more often than not, it's not from looking at the data that we would suspect that. Often the clinical indication is what would lead a clinician to suspect that it might be polygenic. I certainly can't tell by looking at someone's DNA that I think there's multiple there. Certainly we see people with multiple monogenic diseases. That does happen. But yeah, we're still trying to get really good at monogenic disease, and I think we can take that, what we learn there, and apply that to polygenic disease. But certainly I think that will be one of the things of the future that we really need to tackle and get better at, and unfortunately we're just not quite there yet.
And the other thing is, we're reading papers about epigenetics and testing for the epigenetics changes. So I know there are some tests, even articles that have resolved some cases, some diagnosis. Is that something coming soon, or that you probably will be adding to your toolbox?
So we have done a number of epigenetic studies to date, and there's a Canadian project, EpiSign, which is led out of London, that we have collaborated with, and so have helped come up with these epigenetic signatures for a few disorders. We've had patients involved in that, and so continue to collaborate. But certainly that one is led out of London. We're not leading that. We collaborate with them actively and are happy to do so. But yeah, there's a great study on that led out of London.
And just to clarify, because I didn't define...
Sorry, London, Ontario, I should say.
For those who didn't know what I was talking about here, it's because epigenetic changes are not really variants or mutations in the code of the gene, but more in on the DNA, certain signatures on the DNA that either let a gene be expressed or not. So that's epigenetics that we've been hearing more about in the last years. So we do have one question. So you currently use exome sequencing, but the desire to do whole genome sequencing, well, I think that's what you're looking in your new toolbox, I think, the whole genome sequencing. The question is, what is the rate limiting step to move to this technology? Is cost the issue? Is it still investigation and not able to be used clinically?
That's a great question. So yes, cost is one of the main issues. When we look between genome and exome, certainly a genome is much more expensive. There's more sequencing. The other issue is just the quantity of data that we're looking at. And so you can imagine an exome is 1% of your genome. There's a lot less data there, and we know it's enriched for disease mutations. And so the question is really, does it make sense to start with an exome, and if you don't have an answer, reflex to a genome, or do you go straight to a genome? And I think, to be honest, that answer will be a moving target as our sequencing costs keep going down and down. And so right at this moment, that's one of the aims of Activity 4 is to assess genome versus exome in terms of the cost. We are still, I don't have a good answer yet in terms of how many increased diagnoses we get from genome sequencing compared to exome sequencing. We know that we certainly have had a number of great successes in families that we've been looking for answers for years and years. But that's really one of the things that we're looking to do in CARE4RARE is to identify what percent of families can get an answer from genome sequencing versus exome sequencing. And I think what is offered clinically will change as there's these wonderful graphs out there of sequencing costs that's just gone down and down and down over the years. And so at some point we will cross the path to where genome makes more sense. Right now, exome is what's offered. I'll also say the other thing, that it's an interesting balance, because to prepare the lab side of an exome actually takes a lot longer than the lab side of a genome, several days longer, which depending on it adds. And so it's an interesting equation, because there's also lab hours involved in terms of, that's on my side of things what we think of, because cost isn't just the amount to run the sequencer, it's also the people who are preparing the samples and how this works. And then also, as I said, just the quantity of data that you get from a genome, and how do we store that and process that appropriately. So we're working on answering that question, is basically the answer, but right now exome is less expensive, and we can handle that data quite well, and we know that it's certainly enriched for answers in that coding region.
Well, by coincidence, I read my Orphan newsletter this morning, and there was an article about France where they have this new project, Genome Project 2025. I think they want to really reduce delays in diagnosis by using the sequencing technologies, and they have validated that's what I read, but I didn't go into the details. It seems 51 indications where they're doing genome sequencing clinically. They've put it in their clinical panel testing. So, but I didn't have time to look at which are those 51 indications, and so you know, it's advancing also.
There's lots of great studies worldwide actually right now that are comparing exome and genome, and as I said, we're playing in that same sandbox of really trying to understand for our Canadian patients what does the diagnostic rate look like for each technology. But yeah, I think that question is something we continually reevaluate on a yearly basis because the cost of running the tests changes over time.
Yes, that's the positive aspect of sequencing these technologies. So I don't think we're having other questions here. I wanted to, I don't know if, I hope I don't put you on the spot, but I've heard certain cases that you solved at CARE4RARE where it really had an immediate impact on treatment. So, and I guess you have some of those families in mind. Can you describe some of those where finding the gene really helped the treatment and the management?
I think, yes. So it's every once in a while we have a case where it's nice that we can then offer a treatment or inform that treatment. One comes to mind, which is an epilepsy syndrome, that once they were able to identify what this was, we knew that a certain vitamin would help in treating that. And so we're able to help supplement that in those families and try that. And so I will say, unfortunately, I don't have a ton that come to mind of where we were able to offer an actual treatment. We certainly have many, many stories where we're able to help families for family planning purposes or understanding risk of recurrence and things like that. And so I think that's more so we have many, many stories where families come back to us once we've found changes that, for example, are de novo, meaning that it arose in a child, and some of which have particularly tragic stories of deceased children, and knowing that there's very, very little risk for that to happen again because that change just arose in that child. And we've had many very thankful families to know that they could move on to have more children and without worrying about the same thing happening again. And so to be honest, we have more of those stories than of the ones where we have a direct treatment right away. But they're all certainly, it's nice to be able to hear the positive impacts, even for those without a treatment, that it's just had impacts on their family in terms of understanding what the answers are and moving on. But there's been many for knowing families in terms of reassurance of having future children or other grandchildren and whatnot, that there was perhaps not the risk that there could have been once we found the cause.
Yes, and I think you, because we had that researcher who presented 2 days ago from Quebec City, he also finds some de novo variants. Is it, what's the frequencies? Do you find often de novo variants, which would mean that there's no, it wasn't hereditary, it wasn't inherited from the parents as such. Do you find a lot of those, or?
We do. I don't have a number offhand, I would say, between recessives and de novos or dominant, but certainly it's not an infrequent mechanism of disease where a change has arisen in the child that wasn't in the parents.
So doesn't seem to be other questions from our audience. And so just one last question. I was speaking to a mother yesterday who is still looking for a diagnosis, and she's in contact with other families where genes were associated with the children's disease. And I was asking her, for you, because as she was saying, now we we get these acronyms, you know. It's not a name of a disease, it's an acronym. It's the gene name with the mutation with all those letters and numbers. And she knows, because her children are, well, they're young adults now. Actually, she has two. And she said that won't change anything for, you know, for my children, most probably. But I was asking if it still would be a relief or some hope for her. And I think, you know, her answer was, well, I don't think it'll change anything for my family, but yes, the hope, you know, that further research. I guess because the final objective of this is to understand what's happening and to see if they can get some therapies for all these conditions. So I guess that's ultimately what your work is aiming.
Yeah, I think we have to understand what the DNA mutation is and what it's doing before we can really move on to, as a field, develop therapies that are effective. And so I think we're focused on step one of that, of really understanding what these diseases are, and then hopefully from there, groups that are looking at therapies, and hopefully with the ongoing evolution of gene therapy, that really will be able to have some effective treatments for some of these conditions.
Yeah, that's the hope I think families are holding on to. So thank you very much, Kristin, for this very informative presentation. And I just want to briefly tell about our other activities. And tomorrow is first. I think it's a first worldwide, but we're having a day of quiz, a contest for medical students here in Quebec. That part of our activities is only in French, but we have 8 teams from 3 faculties of medicine in Quebec who will be competing, and it's a questionnaire on rare diseases. And again, the aim is that we're not quizzing them on 7,000 rare diseases, but also on what are rare diseases, the impact for the families, the issues, and also what they should think of to think of rare in their career as a physician. So that's going on all day. And next week we have a presentation by Sharon Terry, CEO of Genetic Alliance in the United States, who just started an initiative to be able to some families have access actually to exome sequencing to find some diagnosis. And our last webinar next week will be our life science hub we have here in Quebec, Montreal In Vivo, and RQMO is partner in that hub. And we're presenting a strategic plan for rare diseases, especially research for rare diseases in Quebec, because our government is looking into a rare disease policy and should be announced this year. And finally, we have our Education Day on February 26th, a day where we want to, patients, caregivers, parents, we want them to give their personal stories. We have some testimonials and exchange on how we can reduce delays in diagnosis in the physician's office. How can we bring physicians to think of rare, and also how we can support families without a diagnosis. We will have a presentation that was pre-recorded by SWAN UK, which is an organization, Syndromes Without a Name, that help patients who are undiagnosed. And so that's our upcoming events, and you can get all the information on our website, which is rqmo.org. So thank you again, Kristin, and we hope to hear you in the future with even more good news.
Thank you for having me. Take care.
Bye.
My love.